Хорошо. Давайте проверим, идет, так, проходит, да, движение слайдов проходит. Так, я могу начинать? Да, спасибо, Дмитрий, благодарю вас. Спасибо. Good afternoon, ladies and gentlemen. Topic of today's lecture is immune responses to different types of microbes. What are these microbes? And we'll compare immune responses against bacteria, viruses, fungi, Protozoa organisms, and helminths. Our task would be to characterize distinctive characteristics of these immune responses. So, general features of immunity to microorganisms of different types. First, defense against microbes is mediated by both innate immunity mechanisms and acquired immunity mechanisms. Of course, in real life, they collaborate. Second characteristic, all the immune responses for different microbes are specialized. Specialization of acquired immunity allows the host organism to respond to different types of microbes selectively. For example, synthesis of different isotypes of antibodies, such as immunoglobulin G, M, immunoglobulin A, and immunoglobulin E, may activate protective mechanisms that have the different effects on different microbes. But for example, helminths, they are comparatively they are not sensitive, they are comparatively resistant to immune mechanisms initiated by immunoglobulin G or M. But helminths are very sensitive to responses where important role is the role of the immunoglobulin E. And immunoglobulin E causes inflammation that needs allergy that plays a very important role in the elimination of the helminths from the intestinal tract. Mentioning about cells, the association of cells. Different subclasses of effector CD4 T helper cells play a different role, have different importance in the elimination of different microbes. For example, T helper type 1, a very important helper type 2, and T helper type 17, what responses. First type, second, or 17th type, they important, they have different importance and they're struggling against different types of microbes. For intercellular parasites, most important things are responses supported by T helper type 1 and T helper type 17. For example, for Ect Cellular Bacteria, T helper type 2 immune responses. Third point, if latent or persistent infection is formed, the immunity against this infection may control but cannot eliminate the microbe. Persistent infection means persistence, it means long-lasting surviving of microbe in the human organism without clinical manifestations, but microbe is present in human body. And the last point, in many diseases, tissue injury caused by the host immune responses may be greater than damage caused by the microbe. For example, some infections, Salmonella, for example, may cause bacteriogenic shock or endotoxic shock that may cause the death of the patient. This shock is a direct result of the releasing of pro-inflammatory cytokines by macrophages. It means human products may serve as killers of the human cells and organism. Is innate immunity to Ect Cellular Bacteria. Beginning from this slide, we'll start immunity against extracellular bacteria. Extracellular, it means bacteria that usually live outside of the human cells. For example, Staphylococcus, Streptococcus, Escherichia, Salmonella, and so on. Salmonella may live inside also. So, very important instruments, mechanisms of the struggling against extracellular bacteria are complement activation, we see here, I'm drawing characterizing different pathways of activation: lectin activation pathway, classical pathway, and alternative pathway. All these pathways, when realized completely, causes the formation of membrane complex that causes lysis of the cell. On the surface of this complex is activated. Second, phagocytosis. Phagocytosis may be realized by many types of cells. Phagocytosis is the method of the engulfment, digestion, and killing of bacteria in the phagolysis of the dendritic cells, macrophages, neutrophils, Fungal cells, and some other. And of course, third thing that we must say about is inflammation. Inflammatory reactions are the most important mechanism of the innate immunity with Ect cellular bacteria. These reactions usually involve many mechanisms, including phagocytosis and complement activation. Innate immunity Ect cellular bacteria is based on the recognizing of the bacterial pathogen-associated molecular patterns. Pattern-recognizing receptors, various pattern-recognizing receptors, are present on the surface of our immune cells, and after recognizing of the substances of bacteria that are not typical for humans, they begin signaling of the cell about the irritation. These receptors, pattern-recognizing receptors, are mannose receptors that recognize terminal mannose, L-fucose, the polysaccharides of bacteria. Scavenger receptors, or CD36, of macrophages, recognize and mediate engulfment of lipopolysaccharides, oxidized lipoproteins, and some other microbial products. Third, Fc receptors recognize bacteria opsonized with immunoglobulins, immunoglobulin G, or immunoglobulin M. Fc receptors are very important because they react with the immunoglobulins, and immunoglobulins labeling, making bacteria visible for cells, realize this visualization of bacteria where binding of antibody that before reacted to the antigen to the Fc receptors of the macrophages, neutrophils, dendritic cells, and many other immune cells. Fourth, complement receptors, C3b receptors, recognize bacteria that causes opsonization of bacteria by the C3b portion of the complement. Toll-like receptors of different types, toll-like receptor 1, 2, 3, many. They're important membrane-associated receptors recognizing patterns of microbes, and Minkel receptors. This is translated as the macrophage inducible calcium-dependent C-type lectin. But Minkel receptors recognize pathogen-associated molecular patterns, some carbohydrates of mycobacteria and carinobacteria. In this slide, you see 4 types of receptors that may recognize more than one type of the pathogens. But Dictin-1 receptor, consisting of 2 transmembrane chains. This receptor may recognize pathogen-associated molecular patterns of fungi, they are shown purple, and bacteria, shown green. This is Dictin-1. Dictin-2 react to the bacterial patterns, fungal patterns, and the helmintic substances that are released from the helmintic eggs. It means bacteria, antifungal, antibacterial, anti-helmintic immune responses may be initiated by reaction of the pathogen sheet molecular patterns with the Dictin-2 Minkel receptors. What Minkel? Or do you remember this macrophage inducible calcium-dependent C-type lectin? Mink receptors recognize bacterial and fungal patterns. And last one is blue receptor having only one transmembrane chain. This receptor that reacts to some virus components. And the receptor is named CLEC5A. This is translated as the C-type lectin domain family 5, member A. Now, of course, you must remember this abbreviation, but you have to understand that one of the receptors, what for example this, may recognize and bind to the patterns of the influenza viruses and dengue viruses. In case of the recognizing by this receptor of dengue virus, activation of cells happen, and activated macrophages, for example, but by this recognizing, produce pro-inflammatory cytokines in some patients in very high concentration. These cytokines are tumor necrosis factor alpha, interleukin 1, interleukin 6, interleukin 8, interleukin 17 alpha, interleukin 12, and some others. Exception for this, some chemokines also are released by activated by the fungal pumps inflammation, MIP-A and arantes. What is the result? With all these cytokines and chemokines that are shown here, generate the development of so-called dengue virus-induced hemorrhagic fever. It means systemically in all the circulation of the blood cause the damaging of vessels that cause multiple hemorrhagic reaction. And in the severe cases, this hemorrhagic syndrome may cause dengue shock syndrome that causes death of patients. In this slide, also devoted to the innate immunity extracellular bacteria, you see in the left part of the slide, cytoplasm of the cell, for example, of the macrophage with the brown nucleus. In the right, extracellular space where occur different microorganisms, but bacteria are marked by the red line. Extracellular bacteria may interact with antibodies in the extracellular space and activate complement system that is itself causes inflammation. Bacteria possess molecular patterns may be recognized by mannose receptors, by talc-like receptors of different types, by other receptors, some scavenging receptors. Reaction of bacterial components to these receptors initiate signal, activation signals that by the signaling pathways consisting of many substances with alternative activation by them. Finally, signaling pathway activate nuclear factors that bind to the certain components of the DNA in the chromosomes of the cells, and activate in this way, encoded by these DNAs, pro-inflammatory cytokines released by the cell. For example, interleukin 1 and interleukin 8. These cytokines by themselves activate inflammatory process. And finally, some bacteria via special bacterial injection systems may passage from the outside through the cellular membrane to the cytoplasm and appear in the cytoplasm, damaging cytoplasm of cell from inside. When saying about innate immunity reactions, we have to say that phagocytosis so it's a very important protective mechanism. And phagocytes, such as the dendritic cells and macrophages, and other secrete pro-inflammatory cytokines. You know about this. Interleukin 1, 6, interleukin 23, and other. Accumulation of these cytokines in the infected area causes diapedesis of the migrating the blood vessels, leukocytes, from these vessels to the extravascular tissues where infectious agent is present. This migration of cell is activated by increasing of the vascular permeability that depends on the presence of the listed cytokines. Result is the accumulation, influx and accumulation, so-called recruitment, accumulation of the leukocytes in the affected area. Here you see infiltrate. Practically, this is a pus contain leukocytes. Some of them intensively engulf bacteria that shown inside of the cytoplasm of this cell and some other cells. It means leukocyte infiltration by neutrophils, for example, is a very important element of the inflammatory reaction. And now about Ect Cellular Bacteria and humoral acquired immunity to them. Ect Cellular Bacteria and humoral acquired immunity, acquired adaptive immunity. Humoral acquired immunity response provides a major protection against extracellular bacteria. The central position in this response is the formation of antibodies. B cells, via binding to the bacterial substance, bacterial antigens of the membrane-associated immunoglobulins. You know that this is a immunoglobulin M, monomeric, and immunoglobulin D. But B cells, recognizing specifically antigens of some bacteria, replicate and produce plasma cells. It means they mature to the plasma cells under the control of cytokines of the helper cells. If we say about immunoglobulin response, antibody response, of course, T helpers type 2. Plasma cells produce antibodies. These antibodies neutralize toxin. What in the lower part you see how antibody binds to the toxin molecules, preventing contact of this toxin to the receptors of the host cell. Antibodies cause opsonization. They bind to the bacteria. The Fc fragments becomes recognized by Fc receptors of the phagocytes, and phagocytes very actively engulf the bacteria. Exception for toxin neutralization, opsonization, neutralized antibodies may cause direct clumping of bacteria, forming aggregates of bacteria in the tissues and vessels. This contact activate complement system and help bacteria and help our organisms to eliminate bacteria, killing them. Exception for this, some antibodies may activate complement system, destroying bacteria, and play a role in the antibody-dependent cellular cytotoxicity. This toxicity is lies by natural killer cells that bind by the Fc receptors to the antibodies that called them bacterial cell, the bacterial infected cells, and cause released by these natural killers of the perforin enzymes that may kill human cell and contain intracellular parasites. But this is mostly, of course, mechanism of struggling against intracellular parasites. When seen about T helpers involved in the reaction of the protein antigens of the extracellular bacteria and material of the inflammation and antibody synthesis, we have to say that antibodies mostly are their synthesis is maintained by T helpers type 2. That's not shown in this slide, but exception to T helpers type 2, very important role play T helpers type 17 and T helpers type 1. But in the figure you see upper line of events, the dendritic cell presents antigen to helper cell. This T helper cell is matured to the T helper 17. This mature T helper 17 of this releases cytokines such as interleukin 17, interleukin 22, tumor necrosis after, and some other. These T helper 17 dependent cytokines cause neutrophil inflammation. It means these Th17 cytokines stimulate recruitment of the macrophages and neutrophils and their attraction from vessels to the infected area, infiltration of this area by the cells, and the stimulation of the engulfment of the bacteria by these cells. For macrophage, this is additionally function of antigen presentation. T helper type 1. Usually we say that T helper type 1 producing interferon gamma prevent the development of T helper type 2 immune response. And interleukin 4 secreted by T helper type 2 inhibits maturation of T helpers type 1. But in the real life, T helpers type 1 also play a role in the development of the Ect Cellular Bacteria immune response against Ect Cellular Bacteria by the so-called classical activation of the macrophages. It means in the presence of the interferon gamma produced by T helper type 1 and some other cells, for example, activated natural killer cells, macrophages recruited to the tissue, infected tissue, becomes transformed to the macrophages type 1. They're pro-inflammatory macrophages that are active engulfers of the bacteria. Opsonization for them is very interactive. And these classical activated macrophages produce many pro-inflammatory cytokines. For example, interleukin 1 alpha and beta, interleukin 6, interleukin 12, tumor necrosis factor, and additionally, macrophages very actively produce so-called reactive oxygen species. They are low molecular weight oxygen-containing bactericidal substances that involve in the digestion of bacteria engulfed by macrophages and in the digestion of bacteria outside of the macrophages, because macrophages may release these cytocidal substances to the space surrounding macrophages. So, adaptive immunity to extracellular bacteria mostly is based on the activation of the specific to this bacterial antigen B cells that under control of the cytokines of helper cells are transformed to the plasma cells. Now, after replication, of course, descendants of beta cell to many plasma cells are transformed. And antibodies synthesized by these cells cause neutralization of bacteria, opsonization of bacteria to stimulate the phagocytosis. Antibodies activate complement. In the process of complement activation, influx of the inflammatory cells, neutrophils, for example, macrophages from vessels to the infected areas happen, and altogether they cause the lysis of the cells. But let's comment on this event when T helper cell cytokines stimulate humoral immune response, activating replication of the B cell and transformation of the descendants of the cell to the plasma cells. Bacteria becomes engulfed by dendritic cells. Dendritic cells present this bacterial antigen in the complex with the MHC2 molecule to the T helper cells. To these T helper cells, dependent on the microenvironment, presence of the cytokines in the area where these helper cells, naive T helper cells, are present, may be transformed to different types of T helper cells. If naive Th0 cell is matured to the T helper 17, or this is an upper square, this helper starts producing interleukin 17, 22, tumor necrosis factor. And we know that what these cytokines, cocktail of these cytokines, stimulate inflammation via recruitment of the neutrophils and attraction of the neutrophils from vessels to the inflamed area. If naive T helper 0 is transformed to the T helper type 1, these cells start to produce interferon gamma. We know that interferon gamma is a powerful activator of the macrophages of the first type, pro-inflammatory macrophages, that allows phagocytosis and damaging of infected tissues. And if naive T helpers are transformed to the T helpers type 2, with these cells, T helpers type 2, secreting interleukin 4 and interleukin 4 and 5, activate maturation of B cells to the plasma cells with the production of the immunoglobulins. What 3 types of helpers? Type 17, below T helper 1, below T helper 1, T helper 2. But all these helpers play an important role in the struggle against Ect Cellular Bacteria. Side effects of immune responses to Ect Cellular Bacteria. Immune responses cause inflammation, we understand, and may cause septic shock. Septic shock, this is a dramatic fall of the blood pressure that is associated to the systemic relaxation of the postcapillary veins and opening of the collateral vessels. It means volume, internal volume of the vessels is increased, volume of the blood not. And these things cause the shock when oxygen support of the cells in the many important organs responsible for surviving is weakened. Inflammation is the result of the reaction of the macrophages and neutrophils. These position-associated leukocytes may cause tissue damaging by local production of the reactive oxygen species, nitric oxide or hypochlorous radicals, for example, and by the secretion of cytokines, pro-inflammatory, that stimulate synthesis of fused fat proteins that also participate in the development of the symptoms of the disease, such as the fever, headache, chills, rash, and myalgia. It means what these symptoms appearance depends on the increase of the concentration of the pro-inflammatory cytokines produced by macrophages, some other phagocytotic cells, and production of the acute phase proteins that are stimulated by pro-inflammatory cytokines produced by macrophages. But finally, these inflammatory reactions that may be caused by immune response to this cellular bacteria usually are controlled and self-limited, and usually can cause death, exception for severe cases of the typhoid fever or other septic conditions in the human organism. The next part of the lecture is devoted to the immune responses to intracellular bacteria. Intracellular bacteria can survive within phagocytes because they have different mechanisms of surviving inside of the phagocytotic cell. They're inaccessible to the circulating antibodies, and this is also important mechanism of the bypassing of the immune attack. Intercellular bacteria they require to be eliminated development of the cell-mediated immunity. When not the bacterial cells, but human cells contain bacterial cells inside, must be damaged. And in many infections caused by intercellular bacteria, the host immune response causes tissue injury. Not only extracellular bacteria, but intercellular bacteria also may cause tissue injury. And example of such infection is the tuberculosis. Innate immunity to intracellular bacteria is mediated by phagocytes and natural killer cells. Phagocytes and natural killer cells. Phagocytes ingest and attempt to destroy intracellular bacteria. Initially, neutrophils and after that macrophages try to do this work. But in some cases, these attempts are not effective. Exception for phagocytes, natural killer cells are important in the struggle against intercellular bacteria. These cells kill the host cells infected by intercellular bacteria and by viruses also, because we know that viruses are also intercellular parasites. And we add to this list some intercellular fungi, intercellular protozoa organisms. It means natural killer cells universally may struggle against different intercellular parasites that are not only bacteria but other types also. Activated natural killer cells, exception for destroying of the parasite infected cells, bacteria infected cells, secrete interferon gamma. We know that interferon gamma stimulates macrophages for phagocytotic reactions and for reactions associated with destroying of their own tissues. And interferon gamma stimulates the maturation of naive T helper cells 0 to the T helpers type 1. It means natural killer cell participate in the development and supporting of the cellular immune response. Few words about natural killers. Natural killer cells, they are lymphocyte-like cells that attack the target cell by releasing to the space between natural killer and target cell of the perforins and granzymes. Perforins are shown green. They form the perforations, holes in the cell membrane. Granzymes transported inside through these holes of the cytoplasm, causing heterolytic destroying and damaging of the proteins of the cytosol and nucleic acids of the nucleus. Exception for that, natural killer cells may activate apoptosis in the target cells. You remember that natural killer cells stimulated or non-stimulated due to the balance that is established because between activating signals that activate attack of the natural killer to the target cell and inhibitory signals that shown by the green elements. There are many activating signals that may be expressed on the surface of the infected cell. Usually these substances are bacterial components that appear on the surface of the infected cells, infected by the intercellular bacteria. These substances, for example, may react to the immunoglobulin G antibodies against these substances, and Fc fragments of these immunoglobulins serve as the activating ligands for the activating receptors. What in this case, Fc receptors of the natural killer cell will serve as activating receptors. You see a situation when three units of these signals stimulate cascade of the reactions signaling pathways that activate natural killer for attack. At the same time, if on the surface of the target cell occur on the surface membrane-associated MHC protein type 1, this is a major histocompatibility complex type 1 protein that expresses foreign antigens to the neighboring cells, but this MHC1 protein is itself, independently of what the antigen is expressed, causes activation of the inhibitory receptor, an inhibitory cascade that try to inactivate activating reaction. But in this case, you see that 3 activating signals concur to the only 1 inhibitory signal. Of course, 1 from 3 activating signals would be neutralized, but 2 not. And these 2 signals will finally cause the activation of natural killer to the attack of the infected by the anticellular bacterial cell. Exception for that, natural killer, when is activated by what these activation signals, start producing interferon gamma that stimulate macrophages type 1, to the digestion, active digestion of the bacteria engulfed to the phagocytosis of these macrophages. Adaptive immunity to the intercellular bacteria. So natural killers and phagocytes are cells of the acquired immunity, not of the innate immunity, are adaptive, the cells of the acquired. What the cells of acquired immunity play an important role in the immunity against intercellular bacteria. They are CD40 helper cells and CD8 cytotoxic T lymphocytes. 2 types of cells. It means antigen presenting cell, what here is shown, dendritic cell, if it's infected by intercellular bacteria, and if you have analogous bacteria in the phagocytomes also, presents antigens of the digested bacteria together with MHC2 protein. And in this case, where recognizing of this antigen by the T cell receptor activates CD4 T helper cell, naive CD4 T helper cell. If the C helper cell is stimulated by antigen in the complex with the MHC2 protein by the antigen presenting cell, if this helper cell addition activated by core stimulation signals, for example, CD40 ligand of helper reacts to the CD40 receptor of the dendritic cell, and the CD28 receptor of the co-stimulator receptor of the T helper cell binds to the B7 receptors of the antigen presenting cell. And in this case, if this T helper cell additionally stimulated by interferon gamma, this helper cell stimulated by 3 types of the activating signals, antigen recognizing, co-stimulating recognizing, and humoral activation by interferon gamma, and humoral activation by the interleukin 12 from the side of the antigen presenting cell, with this T helper cell start to be transformed to the T helper type 1. The choice what the helper would be, type helpers would be synthesized, depends on the microenvironment of the cocktail of cytokines that occur in the surrounding space. If naive T helper is present, the prevalent concentration of interferon gamma and interleukin 2, this helper start to be differentiated to helper type 1. This is concerning T cells. No, T helper type 1 produces interferon gamma that additionally enhancing more and more extent of the growth and maturation of the population of CD4 T helper type 1 cells. What is happening with the CD8 lymphocytes? CD8 lymphocytes, precursor cells, if are activated by presentation of the microbial antigen in the complex to MHC1 protein, this is a presentation of endogenous antigen in the contrast to the first with the MHC2. MHC1 presents endogenous antigens. It means antigens of bacteria, viruses that occur in the cytosol. Naive cell activated by the antigen presentation, after that becomes activated by the co-stimulatory signals, what's here. And the third element of activation is activation of this naive cell by interleukin-2 and by interferon gamma. That is not shown here, but we understand that interferon gamma is secreted by T helper type 1. When stimulated by these three types of signals, naive CD8 T0 cell starts its proliferation and form the progeny that is matured this progeny to the differentiated cytotoxic T lymphocytes that have the antigenic specificity analogous to the specificity of this maternal cell, but in the contrast to the maternal cell of this differentiated cytotoxic lymphocytes search for bacteria-infected cells, trying to kill these cells, and from other cells they produce interferon gamma that stimulates macrophages, stimulates maturation of T helper cells and maturation of the naive CD8 cells also. So mediated immunity generated by presence of the anticellular bacteria, anticellular parasites, activation of the macrophage is caused by CD40 ligand binding to the CD40 ligand in the reaction of the co-receptors between dendritic cell or macrophage and T helper cell. This co-stimulation stimulates activation of macrophage and dendritic cells to produce reactive oxygen species. These reactive oxygen species are injected to the phagosomes and fungal lysosomes and activate destroying of bacteria engulfed in these fungal lysosomes. But reactive oxygen species, nitric oxide, hypochloric radical, hydroxyl radicals, H2O2, hydrogen peroxide radicals, and so on, with these substances may act only inside of the phagosomes. But bacteria that are present in the cytosol becomes protected from these reactive oxygen species and survive in the cytoplasm. This production of reactive oxygen species is activated by co-stimulatory signals and by the interferon gamma production. In this scheme, you see events that develop if intercellular bacteria infect phagocytotic cells, for example, macrophages. Example of this bacteria, Mycobacterium tuberculosis or Listeria monocytogenesis, monocytogenes. If macrophage has bacteria in the cytosol, the intercellular bacteria, and in the fungal lysosomes, these macrophages stimulate naive T cells, and if T cells are transformed to the T helper cells, T helper type 1, T helper type 1 cells secrete interferon gamma and tumor necrosis factor that activate digestion of the bacteria in the fungal lysosomes and their elimination in the fungal lysosomes. But interferon gamma and tuberculosis factor presence, they cannot eliminate bacteria from the cytosol. What is happening to eliminate infection in this case? CD8 cells activated by presentation of endogenous antigen with the MHC class 1. This antigen is produced by intercellular bacteria. These naive T cells replicate and transform to the cytotoxic lymphocytes. These lymphocytes recognize bacteria, recognize human cells. For example, this macrophage contain bacterial antigens presented by MHC1 protein of the infected cell, and start this cytotoxic lymphocyte attack against this bacterial infected cell. And all the cell is damaged as a result of this attack, and all the bacteria that occur in this cell are also damaged. In this case, you see complete elimination of microorganisms from human organism, even if this microbe is an intercellular parasite. Immunity to viruses. The next point, viruses. We know that viruses are universal intercellular parasites, but viruses may occur in the form of the intracellular virus and the Ect Cellular variant that may be detected and eliminated via antibodies. So, what are major mechanisms of antivirus protection? Type 1 interferon synthesis. Here you see the dendritic cell that engulfs viruses, and if these viruses enter to the cytosol of this cell, making this cell infected, the cell starts to produce interferon alpha and interferon beta. Interferon alpha and interferon beta are grouped in the so-called type 1 interferons family. This type interferon family bind to the interferon receptors of the human cells, activating synthesis of antivirus proteins in neighboring cells, in macrophages, natural killer cells, some epithelial cells, and other. Natural killer cells. Natural killer cells, when recognize virus antigens presented as the endogenous antigens by the dendritic cells or other cells in the complex with MHC1 protein, this process is not shown, becomes activated, attack this virus-infected cell, and may kill them. Exception for that. Natural killer cells, when activated, secrete interferon gamma. Interferon gamma, you know, activate macrophages by the classical mechanism, making them macrophage type 1 that actively engulf parasites, digest them, and activated macrophages produce interleukin 12, and the dendritic cells also produce interleukin 12. Well, this interleukin 12 participate together with interferon gamma in the transformation of naive T helper cell to the T helper type 1 that play a very important role in the elimination of the virus infection for the human organism. So, protective mechanisms involve secretion of type 1 interferons, activation of the natural killer cells. Both these things are parts of the innate immunity. Immunity to viruses also consists of the synthesis of neutralizing antibodies. This process is not shown here. And formation of cytotoxic lymphocytes that eliminate virus-infected cells. Innate immunity to the viruses is based on the activation of the natural killer cells. Here you see that natural killer cell, when is activated by by what's generation of many activating signals and low number of the inhibitory signals, natural killer cell play a role as the direct destroying of the virus-infected cells and the stimulation of macrophages. In the right part of the slide, you see processes of the activation of interferon system. Left cell shown here becomes infected, but in the process of the replication of virus in the cell, specific signaling pathway activates synthesis of the interferon genes, synthesis of interferons by this infected cell. And these interferons, alpha and beta interferons, are released. These interferons bind to the receptor for membrane non-infected cell and activate synthesis in the cells of the antiviral proteins. These proteins block replication of virus, mostly via the inhibition of synthesis of virus nucleic acids, first of all messenger RNA. And with this, right cell becomes ready to withstand attack of the viruses and prevent the replication of viruses, because here the antivirus proteins synthesized as a result of the signaling via the interferon alpha and interferon beta receptors. Some viruses shut off class 1 MHC1 expression, and in this way escape mechanism from the cytotoxic T lymphocytes. It means we know that natural killer cell may attack target cell if number of activating signals is higher than number of inhibitory signals. Inhibitory signals, this is a signal of recognition of MHC1 protein on the surface of the cell. Some viruses, for example, human immunodeficiency virus, infecting cell, inhibits synthesis of MHC1 proteins by this cell. MHC1 proteins disappear. Why this activity is important for virus? Because only cells infected by the virus and other parasites that have the MHC1 proteins and that present this virus antigen by these MHC1 proteins may be recognized by cytotoxic T lymphocytes. Activation of natural killer cell is different. A natural killer cell becomes activated in the situation when MHC1 protein is absent or mutated, changed chemically. But even this mutation or disappearance of MHC1 protein is one of the instruments of surviving of the viruses in the human cells. It means a virus-infected cell becomes invisible for cytotoxic T cells, but attractive and visible for natural killer cells. It means that natural killer cells and cytotoxic T lymphocytes, CD8 T lymphocytes, they overlap the spectrum of possibilities. All the virus-infected cells that have MHC1 protein are attacked by CD8 cytotoxic T lymphocytes. All the virus-infected cells that have no MHC1 proteins become attacked by the natural killer cells. In the adaptive immunity, now we'll say about adaptive immunity against viruses. In the adaptive immunity to viruses, very important role is the role of the antibodies and cytotoxic lymphocytes. Cytotoxic lymphocytes, they attack virus-infected cells that have on the surface MHC1 proteins. What about neutralization antibodies? Antibodies, when synthesized by the plasma cells after activation of the B cells, they may bind antibodies, bind to the attachment receptors of the virus, preventing attachment of the virus to the host cell. Antibodies may bind to the virus antigens on the surface of the virus-infected cell, making the cell labeled for attack of the phagocytes by the opsonization mechanism, activation of the complement system. In this case, is important role is the role of the Th2 helper cells. So in this case, antigen-presenting cell presents the virus antigen in the complex with MHC class II molecule to the naive T helper cells that in the presence of the prevalent concentration of interleukin 4 and 5 mature to the T helpers type 2, and the T helpers type 2 produce with this interleukin 4 and 5 and stimulate replication of B cells that recognized before antigen, and transformation of them to the plasma cell that secrete high amounts of the antibodies. What else? Adaptive immunity to viruses is mediated by antibodies, exception for cytotoxic T lymphocytes, exception for macrophages, exception for natural killer cells. Role of the antibodies. Antibodies function mainly to prevent initial infection and cell-to-cell spread of the virus. No, cell-to-cell spread, it means virus must bud from the cell and after that infect the next cell. What is this time between budding from first cell infection to the second, virus occurs as the Ect cellular variant. What this variant may be recognized by antibodies, may be bounded to these antibodies, and this way may be neutralized. Exception for immunoglobulin G and M, that what may neutralize viruses inside of the human cell. Secreted immunoglobulin A isotype is important for neutralization of the viruses within the respiratory and the intestinal tract. You remember that secretory immunoglobulin A is in the form of the dimer, is released to the lumen of the respiratory tract, lumen of the intestinal tract, and bind in these secretions to the viruses, preventing the attachment to the epithelial cells of the tracts, respiratory or intestinal. And of course, antibodies may absonate viruses and promote the clearance by phagocytosis and complement activation, clearance of bacteria. Human immunity induced by previous infection or vaccination is able to protect individuals from viral infection, but it's self-eradication. It means if bacteria present in the human organism, viruses is present, and if people have antivirus antibodies when viruses are present, in many cases for a long time balance is established that guarantee occurrence of the protection of human cells and for viruses surviving in this human organism for the long time. No, this scheme is analogous of the scheme of the elimination of the intercellular bacteria. What do you see here? You see activation of the replication of the CD4 cells and their transformation to the matured T helper type 1 cells. It means the elimination of viruses is a very important role of the CD4 T helpers type 1 cells that activate phagocytotic digestion of the viruses and activate formation of the clones of the differentiated cytotoxic T lymphocytes that attack virus-infected cell, causing damage of the cell and parasites that occur inside. Cytotoxic lymphocytes eliminate viruses by killing infected cells, and the dendritic cell in this process may maintain so-called cross-presentation or cross-priming. What is this? If virus infects a tissue cell, not here, blue cell with the virus, this cell usually is not a professional antigen-presenting cell, epithelial cells, for example. But in this case, the infected cell may be phagocytosed by dendritic cell, or macrophages. But now we will discuss problems related to dendritic cell. This dendritic cell, after engulfment of the virus-infected tissue cell, processes this cell and they destroy this cell and processes antigen of this cell. And only dendritic cell, not the macrophage, may present antigens of the virus, in this case, in the complex of the MHC1 molecule, and activate, in this case, naive T killer cell. Antigen-presenting cell also may present virus antigen to the T helper cells, but this process is not shown in the complex with MHC2. It means, in this case, the dendritic cell may remain non-infected, and independently of this may present virus antigens in the complex with the MHC1 protein and in the complex with the MHC2 protein. It means in the complex with the MHC2 protein, virus, so the dendritic cell activates antivirus humoral immune response. If MHC1, if T helper type 1 is activated, cellular immune response. Naive T killer cell, CD8 T killer cell, becomes activated by the MHC1 presentation in the complex with the MHC1 of the virus antigen. Of course, naive cell, after this stimulation, and stimulation by the interferon gamma of the T helper type 1 cell, is differentiated to the cytotoxic lymphocytes, to the population of these lymphocytes that produce interferon gamma, stimulating antigen-presenting cells, and differentiate cytotoxic lymphocytes attack other tissue cells, causing damaging of these cells. What is the peculiarity of this slide? Cross-priming. Why may say about this? But macrophages, for example, may present antigen to the cytotoxic CD8 cells only in the complex with the MHC1 protein, only if macrophage is infected in itself, if virus occurs in the leaf form of this macrophage. But the dendritic cell differ from macrophage, and the dendritic cell may present virus antigen in the complex with MHC1 protein if this virus occurs only in the digested form in the fungal lysosome. This can be done by macrophage, but can be done by dendritic cell. The dendritic cell survives, remains non-infected, but cross-present antigen to cell killer cells and activate cellular immune response. No, this isn't. Few words about different types of the parasites. Fungi. Fungi usually develop in the patient's organism with the immunocompromised situation. It means fungi are typical opportunistic agents that develop when immune response and immune system of host is weakened. Whereas fungi may live in extracellular space and in intercellular also. For example, Histoplasma capsulatum shown here may occur in soil in the form of the molds, but when in fact the human organism is transformed to the yeast form, and this yeast form may survive inside of the human cells, causing an intensive replication of the fungi inside of the human organism. It means the immune response to fungal infection are often based on the combination of responses to extracellular and intracellular bacteria. Neutrophils and macrophages are major innate immunity cells involved in the antifungal response. These cells usually have a variety of different pathogen recognizing receptors, toll-like receptors, Degtin-1, Degtin-2, Minkel, Mannose receptors that activate phagocytotic cells, among them dendritic cells. Upon recognition of fungal through these receptors, the signals result in generation of T helper type 1 and T helper type 17 immune response. What at the level of the acquired immunity, immune responses of T helper type 1 type and T helper 17 type are prevalent. Importance of antibody is low. Neutrophils and macrophages release fungicidal reactive oxygen species and lysosomal enzymes trying to digest fungal organisms. They engulf fungi and try to kill them, but in some cases this becomes impossible. Macrophage activity may be inhibited by some fungi. For example, Cryptococcus neoformans may inhibit the production of tumor necrosis factor and interleukin-12, these pro-inflammatory cytokines, by macrophages. And Cryptococcus neoformans may activate synthesis in T regulatory cells, human T regulatory cells, of the interleukin-10 that acts as the anti-inflammatory cytokine. Interleukin-10 causes immunosuppression. It means that Cryptococcus neoformans may inhibit both inflammatory reaction and cause immunosuppression that prevents replication of all types of the T lymphocytes. When saying about innate immunity to fungi, a very interesting mechanism is formation of so-called neutrophil extracellular traps. Neutrophil extracellular traps, so-called nets. Process of the formation of neutrophil extracellular traps is named netosis. What is happening? When the fungal cells try to engulf too large, if the leukocytes, neutrophils, try to engulf too large fungal organisms, usually more than one group of neutrophils try to engulf this hypha. Of course, these neutrophils, they cannot engulf this fungus, and they release from the cytoplasm cytocidal substances outside to establish contact between the cytocidal substances to the surface of fungal cell. But in some cases, these oxygen reactive species produced by neutrophils are not enough to kill the fungal organism. They actually only partially may destroy fungal structure. More active from them are nitric oxide and hypochlorous radical. What to limit spread of fungi and growth of fungi in this case, neutrophils and some other cells, for example, basophils, they exploit unusual mechanisms, that is, named the formation of the neutrophil extracellular nets. These extracellular nets are shown what here and here, here and here by the arrows as the process of the binding to the surface of the fungal organism of the desoxyribonucleic acid released from the neutrophils to stick DNA to the fungal surface and prevent spread of these viruses, of these fungi, or replication of these fungi. What in the square E law you see? What do you see? You see stained by red surface of the fungal organism, of the fungal hypha. But by the green color is shown elastase of the fungus, elastase of the neutrophil. A DNA of neutrophil is shown blue. And what is happening? When neutrophils are faced to the large fungal organism, they very quickly push out own DNA. This is DNA of the chromosomes of the neutrophils. This DNA has the electric charge that helps the DNA to be attached to all the negatively charged proteins and carbohydrates of the microbes, including fungal organisms. And DNA, this is a largest molecule in the human cells. When it sticks to the surface of fungi, forms the net that covers all the surface of the fungi, and in this net occur neutrophil enzymes destructive for fungi. Elastase, for example, one of them. And what is happened? Well, this fungus becomes coated by the DNA sheeted to elastase. DNA strongly stick to the fungal organism. Elastase destroys this fungal organism, and as a result, fungal organism is destroyed, or at least can grow. No, you know that some receptors are very sensitive to fungal organisms, what dictin receptors, for example. But if dictin receptor reacts to the fungal products of the fungal cell, the DT cell that recognizes the fungus in this way start producing pro-inflammatory cytokines, interleukin 1, 6, 23, and interleukin 12. These cytokines activate replication of naive T cells and their maturation to the T helpers type 17. And T helpers type 17 start producing cocktail of interleukins, interleukin 17, 22, 25, tubular necrosis factor that altogether cause intensive inflammatory reaction. And these cytokines stimulate neutrophils and macrophages to migrate from vessels towards the infected area. It means dictin-1 receptor binding to the fungal products activate process of the formation of T helper 17 immune response in the human organism. And this response causes intensive inflammation and neutrophil inflammation, attracting phagocytotic cells very intensively to the infected area of the human body. Not only dictin-1 may cause this effect. During the reaction against intracellular extracellular fungi, the dendritic cells produce cytokines stimulating T helper type 17 immune response. We talked about this. This may be stimulated by the soluble fungal elements by the Ect Cellular fungi or fungi that occur in the apoptotized cells. In this scheme, you see some events that occur when fungi infect human organisms. Now, in the left upper part, you see macrophage that phagocytosis is fungal organisms. This macrophage digests fungi and produce pro-inflammatory interleukin-1, beta, tumor necrosis factor, and leukin-6. These cytokines stimulate inflammatory reaction, and they stimulate maturation of population of T helper type 17 that is shown in the right part of the slide. T helper type 17 produce cytokines, for example, interleukin-22, that stimulate synthesis in the epithelial cells of the beta defensins that destroy fungal organisms in the secretions. What else? Pro-inflammatory cytokines and cytokines of the T helper type 17, interleukin-17, they cause the influx, so migration from the blood of the neutrophils to the infected area. We see them in the central part of this slide. And neutrophils, after recognizing of fungal pumps by the receptor neutrophils, they cause netosis. It means they push out the DNase and elastase and immobilize fungal cells and destroy fungal cells trapped in these neutrophil cellular traps. Additionally, natural killer cells migrate, well, this is a left lower part of the slide, from the vessel to the tissues, produce interferon gamma. That's not shown, but interferon gamma stimulates macrophages for phagocytotic activity and for pro-inflammatory activity. Well, this scheme demonstrating activity of macrophage, natural killer cell by itself may destroy fungal organism or fungus-infected host cell. Summary of mediated fungi. Cell-mediated immune responses against fungi, innate and acquired, is the principal mechanism of immunity. Neutrophils and macrophages initiate the response, including formation of the neutrophils of the nets. Strong T helper 17 responses are generated by some extracellular fungi. They depended on dendritic cells activation by fungal glucans that bind to the dectin-1 receptor. It means T helper 17 immune response has many possibilities to eliminate both extracellular and intercellular fungi, and one of these mechanisms of elimination, this is a stimulation of the attraction of neutrophils to the area of the infection in the human body infected, and formation of the traps. Exception for that, T helper type 1 immune response also is responding cytotoxic lymphocytes that attack the fungal infected cells and eliminate them. Role the antibodies may be important, but this importance is not very high. Importance of immunoglobulin A to prevent attachment of fungi to the surface of epithelial cells. In case of the immunoglobulin E is prevalent, this presence is considered as a signs of the negative situation in the immune response to fungi. Protozoa organisms. No, I have some limited time, and due to this, I have to see. Protozoa organisms mostly are intercellular parasites and cause chronic diseases maintained by the hydrolytic enzymes of these parasites. These parasites, many of them reside intercellularly. What here you see presence of the Leishmania in the leukocyte of humans, Leishmania donovani. In the recognizing of fungal components, very important role of the different toll-like receptors, depending on the stimulation of the immune response by binding to different toll-like receptors. Pro-inflammatory cytokines may be produced by antigen-presenting cells, or inhibitory signals, including interleukin-10 and transforming growth factor beta that inhibit immune responses to parasites. What balance between activation and inhibition of inflammation is typical for many parasitic diseases. Protective mechanisms of immunity are similar to antifungal immunity, are similar to those emitting viruses and intercellular bacteria. First of all, the leading important characteristics of immune responses have T helper type 1 and T helper type 1 response with the synthesis of interferon gamma. T helper type 1 response activates macrophages and cytotoxic CD4 cells that may destroy protozoa infected cell. Macrophages may destroy Protozoa organism directly. Antibody responses. Antibody responses may be important, but importance of antibody is lower. The importance of macrophage activation of T helper type 1 cells and cytotoxic lymphocytes. It means mostly you see immune responses protective against parasites are associated to cellular Th1 immune response. And a few words about helminths. Helminths, in the contrast to all other pathogens that we described before, are very large organisms. For example, moths are too large to be phagocytosed effectively. For example, ova of the Schistosoma mansoni, what here you see this blood fluke, so-called Schistosoma mansoni, size are about 140 micrometers long. And due to this, many worms are resistant to the cytocidal mechanism of macrophages and neutrophils, because this mechanism cannot digest thick tegument of the worms. Tegument is the outer body covering, or like a calyx of the helminths, consisting of proteins, lipids, carbohydrates, and nucleic acids. It forms the protective layer of the worms, surviving both secretory and serving both secretory and absorptive processes. Acquired immunity, non-acquired immunity to fungal organisms has a limited capacity. So the acquired immune response is associated to immunoglobulin E synthesis and involvement in the process of the eosinophils, mast cells, and basophils. Principle, for example, protective mechanism of immune responses to schistosoma species of Schistosoma mansoni is killing of this organism by eosinophils and macrophages. Macrophages by themselves cannot kill, but in the context with the eosinophils can. The principal protective mechanism in response to filaria is cell-mediated immunity. Other organisms. Eosinophil activation and production of immunoglobulin E antibodies is the principal protective mechanism of the immunity against infections caused by different worms. These events are mediated by cytokines derived from T helper type 2. It means prevalent immune response is cellular immune response mediated by Th2, but most important is T helper type 2 cells response, T helper response type 2, associated to secretion interleukin 4 and 5. Well, this interleukin 4 and interleukin 5 are major mediators of protective response. parasitic worms stimulate differentiation of naive CD4 cells to the T helper type 2 because they immune response need to be associated to interleukin 4 and 5. Interleukin 4 stimulates production of immunoglobulin E in the plasma cells, and interleukin 5 stimulates replication of precursor of eosinophils and recruitment of the eosinophils to the blood circulation. What here you see scheme? Eosinophil activation and production of immunoglobulin E, eosin activation, production of immunoglobulin E. These processes are inside of the zone, marked by the red line. They are basic protective mechanisms against worms. Central cell is the T helper type 2. Worms stimulate T helper type 2. T helper type 2 population produces interleukin 4 and 5 that stimulates maturation of eosinophils. They are most killer cells for fungi. And additionally, immunoglobulin E on the surface of the mast cells react to the fungal allergens and cause degranulation of mast cells with the releasing of the histamine. Histamine causes inflammatory reaction, and on the basis of this reaction, eosinophils are attracted to the zone of presence of worm. T helper type 1 and T helper 17th immune responses have the lower importance. What is happening? If basophil here, if eosinophil, the eosinophil is shown that has a C receptor for immunoglobulin E, binds to the immunoglobulin E, and these immunoglobulins bind to the worm antigens, cross reaction of the FC portions of the FC receptors of eosinophil is happening. Well, this cross-linking of FC receptor via immunoglobulin E and via their binding to the worm. This cross-linking of the FC receptors of eosinophils causes the degranulation of eosinophils. And from eosinophils, eosinophil peroxidase and toxic other proteins such as the lysosomal hydrolysis and the major basic protein are released. Why even eosinophils are important? Because eosinophilic peroxidase is distinct from the male peroxidase, the peroxidase of the macrophages or neutrophils. Eosinophil peroxidase promotes synthesis of very highly destructive for worms of the hypochlorous and hypobromous acids. But these hypochlorous and hypobromous acids may destroy tissues of the fungi of the worm and kill this worm organism. Process is named degranulation. Exception for eosinophilic peroxidase. Eosinophils produce pro-inflammatory substances such as the prostaglandins and leukotrienes that maintain chronic inflammation in the zone infected by the worms. The time of the lecture is finished. Thank you for coming to the lecture. Remember, please, that worms are controlled by the eosinophils and immunoglobulin E synthesis. Now, this is a distinctive characteristic of immune response against worms. Goodbye, gentlemen.